My name's Talsma. I'm a research assistant, or was a research assistant at the University of Pittsburgh, where I worked with Dr. Michael Palladino. His lab studies fruit fly models of neurodegeneration, and over the course of his career, he's discovered a number of sodium-potassium pump mutations. So in humans, the sodium-potassium pump mutations cause both hemiplegic migraine type 2 and alternating hemiplegia of childhood and rapid onset dystonia Parkinsonism. There's a big variability in terms of the disease phenotypes and what they look like in human patients, as well as what our flies look like in the lab. And so given this, we decided to do a genetic screen of the entire Drosophila genome to look for other proteins that might interact with the sodium-potassium pump and make the phenotype of the flies worse or better. And the overall goal would be then, if we could find a protein in a fly that makes the phenotype better, we could maybe then look in humans and see if we could modify that protein as well to help human patients. And so we performed a screen and looked at most of the genome and were able to find 50 genes that possibly modify the phenotype of mutant flies. And so it was pretty exciting, and a lot of these proteins affected synaptic development and neuron growth. And so we looked further, and we also, so the second thing that we did is we generated mutant flies that express human mutations in fly cells. And so given that we saw a lot of defects in neuron growth, we also looked at the growth of neurons in flies, and we saw that there were also defects. And so this gives a lot of possible sort of ideas for the future. One is perhaps AHC is caused by some underlying growth defect in neurons, and this growth defect may be localized to a certain part of the brain, and that's why you see more involvement of certain parts of the brain, and also why you don't see general epileptic type seizures. The other interesting thing to look at would be second mutations in AHC and RDP patients to see if that any of those second site mutations are in the same proteins that we identified in our screen, and see if those would potentially be therapeutic targets later on. They've already done a lot of sequencing on AHC patients. The data is probably already there to find mutations in second site genes. And all it would take would be a comparison between the list of genes that our lab generated and common second site mutations in AHC patients. So that step could be relatively quick, but then looking at these genes as potential therapeutic targets would be a lot harder. It would sort of depend on if there's already a drug existing that is known to target these genes and modulate their activity. So unfortunately, I've moved on from the AHC lab, and really what I would like to be able to do is give my data to one of the clinicians and have them use it in their work. And maybe, we heard about there's a number of patients still that have AHC phenotypes but don't have an ATP1A3 mutation. And it would be interesting to see if these patients had maybe mutations in any of the genes that we had, or things like that. So essentially, I would like others to use my data. I'm unfortunately moving on. Yes, if anyone's interested in any of these genes, they should contact me. I have a list, and I can send it to them very easily. There's also a publication that they can look at. Yeah, it's so the list is actually open access on this journal. The journal that has the publication is free, so you can read the article for free and see the list for free. And it's all out there. If you search for, Talsma, Palladino, Drosophila, you should find it.